Welcome back, everybody. Ready to dive in and get microscopic? Today, we're going to be exploring the world of microbial genetics.
Should be a fun one.
Yeah, it's so amazing how these tiny organisms manage their genetic information.
Right.
And we've got some awesome material to help us break it all down. You guys sent in some excerpts from OpenStax's Microbiology Q&A.
Oh, nice.
And looks like the questions focus on mechanisms of microbial genetics. I'm excited.
Yeah, this is a really cool area.
It is. It's super cool. So
Yeah.
get ready to learn about how bacteria replicate their DNA, how they deal with mutations, and even how they swap genes with each other.
Yeah, it's almost like their own little microscopic trading network.
Oh, I love that. Okay, so where should we start? Maybe we should start with the central dogma of molecular biology.
That's always a good place to start, I think.
It's like the genetic information highway, right?
Exactly. It describes how the information flows from the DNA to RNA to protein.
Okay.
So the DNA is like the blueprint. With all the instructions for building and running the cell, RNA is kind of like the messenger,
Okay.
carrying those instructions over to the ribosomes where the proteins are made, and then the proteins are really the workers,
Right.
carrying out the instructions and doing all the jobs in the cell.
Okay, that makes sense. That's a good overview. But how does all of this play out in bacteria? I mean, they're a lot simpler than our cells, right?
Yeah, absolutely. They are much simpler, but the basic principles are pretty much the same.
Oh, okay.
So let's take DNA replication, for example. Bacteria usually have a single circular chromosome,
Okay.
and they need to replicate that chromosome really quickly and accurately before they can divide.
Right. Makes sense. So how do they do it?
Where?
Is it similar to how our cells replicate their DNA?
Yeah, it's similar in a lot of ways, but there's also some really important differences.
Okay.
So both bacteria and our cells use what's called a semi-conservative replication process,
Semi-conservative.
where each strand of the DNA molecule basically serves as a template for building a new complementary strand.
So it's like each new DNA molecule has one old strand and one new strand.
Exactly, you got it.
So are there any unique challenges to this, especially when it comes to bacteria with their circular chromosomes?
You know, that's a good point, because if you imagine, it's kind of like trying to copy a blueprint that's rolled up like a scroll.
Oh, yeah.
It gets a little tricky.
For sure, yeah.
But bacteria have developed some pretty clever solutions to this problem. So one key player here is DNA gyrase.
Okay.
And this enzyme basically helps relieve the tension that builds up as that DNA molecule unwinds for replication.
So almost like a molecular stress ball.
Exactly, yeah. It keeps everything from getting tangled up.
Right. So what are some of the other players on this replication team?
Well, we've got helicase,
Okay.
which, you know what that does.
Unzips the strands.
Unzips the two strands of the DNA molecule.
Yes.
We've got primus, which lays down these short RNA primers
Yeah.
to kind of give DNA polymerase a starting point.
Right.
And then, of course, we've got DNA polymerase, which really does all the heavy lifting in adding new nucleotides to that growing DNA strand.
It's like a construction worker.
Exactly, yeah, putting all the building blocks together. And then last but not least, we've got ligase,
Okay.
which comes in and seals up any gaps in the newly synthesized DNA just to make sure it's a complete and continuous molecule.
So it's like the glue that holds it all together.
Exactly. You got it.
So it sounds like this is a really well-coordinated dance
It is. It is.
with all these different enzymes working together.
It's a beautiful thing.
But I remember reading that there's a difference in how the two strands of DNA are replicated.
You're right. You're right. So there's one strand called the leading strand,
Okay.
and it's synthesized continuously.
Okay.
But then the other strand called the lagging strand is synthesized in these short fragments called Okazaki fragments.
Okazaki fragments. Why is that? Why can't both strands just be replicated the same way?
Well, you see, it all comes down to the directionality of DNA synthesis. So DNA polymerase can only add new nucleotides to what's called the 3' end of a growing DNA strand.
Okay.
And since the two strands of the DNA molecule run anti-parallel to each other,
Yeah.
one strand can be synthesized continuously in the direction of that replication fork, but the other strand has to be synthesized kind of in the opposite direction in these little short pieces.
Oh, that's interesting. So it's almost like trying to write on a scroll that's unrolling?
Yeah, exactly. A great analogy. You can only write in one direction.
Okay. So once the DNA has been replicated, what happens next? How does all of that genetic information get put into action?
Well, that brings us to RNA transcription,
Okay.
which is the process of making an RNA copy of a gene from that DNA blueprint.
So it's kind of like making a photocopy of a specific page.
Yes.
in the instruction manual, just the one that's needed at that moment.
Exactly. And so this RNA copy is called messenger RNA.
Messenger RNA.
Or mRNA for short. And it carries those genetic instructions out of the nucleode region in bacteria to the ribosomes where protein synthesis takes place.
Okay, got it.
Yeah.
So then RNA polymerase is like the copy machine.
Another good analogy. RNA polymerase binds to a specific region of the DNA called the promoter,
Okay.
which signals the start of a gene. And then it unwinds a short section of the DNA and uses one of those DNA strands as a template to build a complementary RNA molecule.
So how does it know when to stop? Are there stop signs in the DNA too?
You know there are. There are specific DNA sequences that act as termination signals,
Oh.
telling the RNA polymerase to detach and release that newly made mRNA molecule.
So it's like having start and stop codons for transcription.
Exactly. Very similar concept.
Okay, so now this mRNA molecule is ready to head to the ribosome for translation, right?
Well, almost.
Almost.
In bacteria, yeah, it's pretty much ready to go, but in eukaryotic cells like ours,
Uh-huh.
that mRNA molecule needs to go through some extra processing before it can leave the nucleus.
Oh, so it's like getting a passport and visa before traveling to another country.
I love that. That's a great way to put it. So eukaryotic mRNA gets what's called a 5' cap and a poly-A tail added,
Okay,
which help protect it from degradation and also assist in its export from the nucleus.
gotcha. So bacterial mRNA is like a domestic flight, while eukaryotic mRNA is like an international flight.
Exactly. It requires a little more preparation. But once that mRNA molecule reaches the ribosome, the process of translation is similar in both bacteria and eukaryotes, right?
Right.
So the ribosome reads that mRNA sequence and uses it to assemble a chain of amino acids in the correct order.
Okay.
And then that chain of amino acids folds into a functional protein.
So we've gone from DNA to RNA to protein. That's the central dogma in action.
You got it.
But I am curious about what happens if there are like errors in the DNA sequence. Don't those errors lead to problems?
They can, you're right.
Yeah.
But sometimes they can actually be beneficial.
What?
Yeah.
Interesting.
We call these errors mutations. They're simply changes in that DNA sequence.
So mutations are like typos in the genetic code.
Exactly. Great way to think about it. Some typos might be really minor
Right.
and not really affect the meaning, but other typos can completely change the message.
Yeah, that makes sense.
And so the consequences of a mutation really depend on where it occurs in that DNA sequence and what kind of change it is.
Gotcha.
Some mutations can be what we call silent,
Okay.
meaning they don't actually change the amino acid sequence of the protein.
Okay.
Others might be missense mutations,
Okay.
where a different amino acid is incorporated, and that could potentially alter the function of the protein.
And then there's also nonsense mutations.
Yes.
Which I imagine can be pretty disruptive.
Yes, they are. They basically introduce a premature stop codon,
Okay.
cutting the protein short,
Okay.
and often rendering it non-functional.
Yeah, so it just stops reading it.
Exactly, yeah. And we can't forget about frame shift mutations.
Right. Frame shift.
Which occur when nucleotides are inserted or deleted
Okay.
in a number that's not divisible by 3.
Okay.
This disrupts the reading frame and completely alters the amino acid sequence downstream of that mutation.
So it's kind of like shifting all the letters in a sentence by one position.
Yes.
It completely changes the meaning.
Exactly. So you can see how a seemingly small change in a bacterium's DNA could, for example, suddenly give it resistance to an antibiotic.
Oh, wow.
Right? And then suddenly that bacterium has a survival advantage.
Right.
And then that resistance gene can spread through the population.
Yeah, that's a scary thought.
That's why understanding these mutations is so crucial for tackling antibiotic resistance.
For sure. It's amazing how such a tiny typo can have such big implications.
It is incredible.
But mutations aren't the only way that bacteria can change their genetic makeup, right?
That's right. They've got some sneaky ways of sharing genes with each other.
Yeah, I was just thinking about that.
It's like they've got their own little genetic trading network.
I'm picturing bacteria swapping genes like trading cards.
Yeah, it's a great visual. So how does all this genetic trading work? Well, bacteria have evolved several mechanisms for what we call horizontal gene transfer.
Horizontal gene transfer?
Which allows them to share genetic information with each other.
Okay.
And there's 3 main ways they do this: transformation, transduction, and conjugation.
Okay, let's break those down a little bit. What's transformation?
So transformation is when bacteria basically take up naked DNA from their surroundings,
Okay.
often from dead cells. So it's kind of like finding a discarded blueprint
Okay.
and incorporating it into your own collection.
So it's like genetic recycling.
Exactly. Yeah.
Okay. What about transduction? Does that involve viruses?
You're good. You're right.
Yes.
So transduction involves these viruses called bacteriophages,
Okay.
and they act as unwitting genetic couriers.
Oh, wow.
So during their life cycle, these phages can accidentally package bacterial DNA
Okay.
into their viral particles and then transfer that DNA to other bacteria upon infection.
So it's like a genetic hijacking.
Exactly. Yeah. So the phage accidentally picks up some bacterial DNA
Right.
and delivers it to a new host.
And then what about conjugation? That's the one where there's like a little bacterial romance going on.
A little bit of bacterial romance.
Yeah.
So it does involve a close encounter between two bacterial cells.
Okay.
And during conjugation, a bacterium carrying a special plasmid called the F plasmid
The F plasmid.
forms a physical connection with another bacterium that doesn't have the plasmid.
Okay.
And this allows for the transfer of that plasmid and sometimes even chromosomal DNA from the donor to the recipient.
So it's like a direct line for sharing genetic information.
Exactly. And I know, this can have significant implications.
Oh, yeah.
Especially when it comes to antibiotic resistance.
Right.
Because conjugation is a major player in the spread of antibiotic resistance genes.
Yeah.
So these R plasmids, which carry genes for antibiotic resistance, can be readily transferred between bacteria through conjugation,
Okay.
which contributes to the growing challenge of treating bacterial infections.
So it's almost like a bacterial arms race.
Well, it is.
With bacteria sharing their resistance genes to try to outsmart the antibiotics.
It's a constant battle.
Yeah. And before we move on from this whole bacterial gene swapping discussion, I want to talk about these fascinating jumping genes called transposons.
Yeah. Transposons are these mobile genetic elements that can hop around within a genome, inserting themselves into different locations. They're like little genetic nomads,
I like that.
traveling from one place to another, and they can even carry genes with them,
Oh, wow.
including genes for virulence factors and antibiotic resistance, which of course contributes to the spread of these traits.
So they're like little genetic hitchhikers,
Exactly.
carrying important cargo from one part of the genome to another.
You got it.
Wow. Okay, so with all this talk of genes moving around and being shared, it seems like gene regulation is super important for maintaining order
It is.
and making sure that the genes are expressed at the right time.
It is super important. Gene regulation is like this complex control panel,
Right.
ensuring that genes are turned on or off as needed.
Okay.
And in prokaryotes, a key regulatory mechanism involves what we call operons,
Operons.
which are clusters of genes under the control of a single promoter.
So they're like teams of genes all working together, responding to the same signals.
A great way to put it, yeah.
Okay. Can you give us some examples of how these operons work? I'm particularly curious about the lac operon
Oh, yeah, the lac operon.
and how it regulates the breakdown of lactose.
Sure. So the lac operon is a classic example of what we call an inducible operon.
An inducible operon.
So it controls the breakdown of lactose, which is a sugar.
Okay.
And when lactose is present,
Mm-hmm.
it binds to a repressor protein, causing it to detach from what's called the operator region and allowing transcription of the genes needed to break down lactose.
So the lac operon is like a switch that gets flipped on when lactose is available,
Exactly.
allowing the cell to use this sugar as a food source.
Yeah.
What happens when lactose isn't present? Is the operon just kind of sitting there idle?
Not exactly. Even when lactose isn't around, there's a low level of transcription of the lac operon genes,
Okay.
just to ensure that the cell has a few lactose processing enzymes ready to go, just in case lactose becomes available. It's kind of like having a pilot light on a stove,
Oh, okay.
ready to ignite the burner when needed.
That's cool. But what about glucose? I know that bacteria prefer glucose as their primary energy source.
Right.
Does glucose affect the lac operon at all?
It does. The lac operon is also subject to what we call catabolite repression,
Metabolite repression.
meaning that it's inhibited by the presence of glucose, which is the cell's preferred sugar. So even if lactose is present, if glucose is also available, the lac operon will be mostly turned off.
So it's like the lac operon is a picky eater, preferring lactose only if glucose isn't on the menu.
Exactly. It only wants the lactose if the glucose isn't around.
It's fascinating how all these regulatory mechanisms work together to make sure that bacteria are using their resources efficiently.
It is, and it's super important for their survival.
Right, yeah.
So this is just one example of the many intricate ways that bacteria control their gene expression. But now I think it's time to put your knowledge to the test.
Okay.
You've provided a set of test questions for us.
I have. I'm ready.
So let's see how well you've absorbed all this information.
All right, working on the challenge. I'm ready to show off my microbial genetics prowess.
All right, I love it. I love the enthusiasm. So let's start with a multiple choice question just to warm up your brain.
Okay.
All right, here's the first one. DNA does all but which of the following?
Okay.
And your options are: AR serves as the genetic material passed from parent to offspring. B provides the instructions for the synthesis of messenger RNA.
Okay.
C remains constant despite changes in environmental conditions. Or D is read by ribosomes during the process of translation.
Okay, let me think about this for a second. So we've talked about how DNA is like the blueprint,
Right.
the master copy of the genetic information.
Yes.
And it definitely gets passed down from one generation to the next. It also provides the instructions for making mRNA, which then carries those instructions to the ribosomes.
Right.
And DNA itself doesn't change in response to the environment, though which genes are expressed can certainly vary.
You're on the right track.
Okay.
So which option doesn't quite fit with the role of DNA?
I think it's D.
Okay.
Is read by ribosomes during the process of translation. Because DNA doesn't go to the ribosomes. It's the mRNA that gets translated into protein.
You are absolutely correct.
Yeah.
You have correctly identified the function that does not belong to DNA.
Awesome. Okay, I'm ready for the next challenge.
All right, I love it. Here's a true or false question.
It won't trip you up.
Okay.
Cells are always producing proteins from every gene they possess. What do you think?
Oh, that's a tricky one. I know that cells have tons of genes, but I don't think it makes sense for them to be expressing all of them all the time. That would be such a waste of energy.
That's a great point. You got to be efficient, right?
Right.
Cells need to be selective about which genes they express depending on what they need and their environmental conditions.
So the statement is false.
That's right.
Cells are not always producing proteins from every gene they possess.
Exactly. They regulate that gene expression very carefully to make sure they're only making what they need when they need it.
It's like a chef using a recipe book.
Yes.
They don't make every dish in the book all at once.
That's right.
They pick the recipe based on what ingredients they have and who they're cooking for.
Perfect analogy.
All right. I'm ready for another question. Let's see what you've got.
All right. Let's test your understanding of mutations a little bit more with this one.
Which of the following is a change in the sequence that leads to the formation of a stop codon?
Okay.
And your options are a silent mutation,
Okay.
B, nonsense mutation,
Okay.
C, missense mutation,
Okay.
or D, frameshift mutation.
All right. So I remember we talked about the different types of mutations and how they can affect that protein sequence. So silent mutations don't change the amino acid sequence, so they can't really introduce a stop codon.
Right.
Missense mutations change one amino acid to another, but they don't necessarily create a stopped codon.
Right.
And frameshift mutations can really mess things up by shifting that reading frame,
Right.
but they don't always lead to a stop codon either.
You're really thinking this through.
So which type of mutation is the most likely to introduce that premature stop codon?
What do you think?
I think it's B, a nonsense mutation.
You are correct.
Yes.
It's the one that changes a codon that codes for an amino acid into a stop codon, prematurely terminating that translation.
I feel like a mutation detective. Okay, what's our next case?
All right, let's delve into the world of horizontal gene transfer with this one.
Good.
It's a fill-in-the-blank question. The F plasmid is involved in which of the following processes?
Oh, the F plasmid. That's the one that lets bacteria connect and transfer genetic material during conjugation. So the answer is conjugation.
Yes, you got it.
Awesome.
The F plasmid is the key player in bacterial conjugation, enabling them to share those genes and spread traits like antibiotic resistance.
It's amazing how these tiny organisms have evolved such sophisticated ways to exchange genetic information.
It really is. It's quite impressive. All right, ready for another one?
Yes, absolutely.
Let's switch gears just a bit
Okay.
and test your knowledge of operon regulation
Right.
with this short answer question. Describe the regulation of the TREP operon and how it differs from the regulation of the LAC operon.
Okay, this one requires a little more thought.
All right, take your time.
So both the TRP operon and the LAC operon are examples of how bacteria control the expression of genes involved in metabolic pathways.
Right.
But they use kind of different strategies.
Mm-hmm.
So the TRP operon, which controls the synthesis of the amino acid tryptophan, is a repressible operon.
You're on the right track. Can you elaborate on what makes it repressible?
Well, so when tryptophan is abundant,
You're right.
it binds to a repressor protein, which then binds to the operator region of the operon, blocking transcription of the genes involved in tryptophan synthesis. So basically, the presence of tryptophan shuts off the operon.
That's a perfect explanation. It's like a feedback mechanism,
Right.
ensuring that the cell only makes tryptophan when it needs it.
Makes sense.
How does that differ from the regulation of that lac operon?
Well, the lac operon, which controls the breakdown of lactose, is an inducible operon.
Okay.
So it's turned on in the presence of lactose. Lactose binds to the repressor protein, causing it to detach from that operator region and allowing transcription of the genes needed to break down lactose.
Exactly.
So the TREP operon is turned off by the presence of its end product,
Mm-hmm.
while the lac operon is turned on by the presence of its substrate.
You've got it. It's fantastic how bacteria have evolved these different regulatory strategies to fine-tune gene expression and optimize their resource use.
Yeah, they're so efficient.
They are. And these intricate mechanisms, it's just a small glimpse into the amazing world of microbial genetics. We've covered so much ground today.
We have.
We talked about DNA replication, transcription, translation, mutations, horizontal gene transfer, and gene regulation.
A lot.
I'm so impressed you've demonstrated a solid understanding of all the fundamental principles and tackled some challenging questions.
I feel like I aced that pop quiz. I've learned so much from this deep dive.
Oh, good. I'm glad. It's incredible how much complexity there is packed into these tiny organisms.
Oh, no, right.
So much going on. But for now, I think we should take a break. And then we can come back and explore a few more questions that will really make you think about the broader implications of all this microbial genetics.
I can't wait.
Welcome back to our deep dive into microbial genetics. Are you ready to tackle some more challenging questions?
Absolutely. I'm feeling energized and ready to push my understanding even further.
Okay, here's one that requires you to think about the interplay between genotype and phenotype. Can two observably different cells have the same genotype? Explain.
I think the answer is yes. Two cells can have the same genotype but different phenotypes. The environment can play a huge role in how genes are expressed.
Go.
Like, for example, imagine two identical twin bacteria, both carrying the gene for producing a pigment that makes them appear red.
Okay.
If one twin is grown in a nutrient-rich environment, it might produce lots of pigment and be bright red. But if the other twin is grown in a nutrient-poor environment, it might not produce as much pigment and appear pale pink.
That's a great example. So even though the twins have the same genetic makeup, their appearance or phenotype can vary depending on their environment. It's like two people with the same genes for height. One might grow taller if they have a healthy diet and plenty of exercise, while the other might be shorter if they don't have access to those same resources.
So the genotype is like the blueprint, but the phenotype is the actual building, and the environment can influence the construction process.
That's a great analogy. Now let's move on to a question that focuses on a key enzyme in DNA replication. Why is primus required for DNA replication?
Okay, primus is the enzyme that makes those short RNA primers, right?
Right.
I remember that DNA polymerase can't just start building a new DNA strand from scratch. It needs a starting point, a free 3-foot hydroxyl group to attach the first nucleotide to.
Exactly, and that's where primus comes in.
Right.
It provides that initial 3-foot OH group by laying down a short RNA primer, giving DNA polymerase a foundation to build upon.
So it's like primus is the construction crew that lays the foundation, allowing DNA polymerase to come in and build the rest of the house.
I like that analogy. Now let's shift gears and think about a specific type of mutation. Describe what happens when a nonsense mutation is introduced into the gene encoding transposase within a transposon.
Okay, transposase is the enzyme that allows transposons to jump around in the genome, right?
Right.
And a nonsense mutation introduces a premature stop codon, which can really mess up a protein.
It can. What do you think would happen if a nonsense mutation occurred in that transposase gene?
Well, if the transposase gene is truncated and non-functional, the transposon wouldn't be able to move. It would be stuck in its current location.
That's precisely right. The transposon would be like a car with a broken engine, unable to move to a new location.
So those jumping genes would be grounded. I'm starting to appreciate how even small changes in DNA sequence can have a big impact on cellular processes.
Absolutely. And as we wrap up our deep dive into microbial genetics, I want to leave you with a final thought-provoking question. We've focused on bacteria, but how do you think gene regulation might work differently in more complex organisms like us?
That's a great question. I know that eukaryotic cells have a nucleus, which separates transcription and translation,
Right.
and their DNA is packaged with histones forming chromatin.
Mm-hmm.
So I'm guessing that these features add layers of complexity to gene regulation. maybe there are more opportunities for fine-tuning gene expression in eukaryotes.
You're on the right track. Eukaryotic gene regulation is a whole other fascinating world waiting to be explored. But for now, I want to thank you for joining me on this deep dive into the amazing world of microbial genetics. You've demonstrated a keen understanding of the key concepts and a genuine curiosity about the intricate workings of these microscopic wonders.
Thank you so much for guiding me through this fascinating landscape. I've learned so much about how bacteria manage their genetic information, from DNA replication to gene regulation. And I'm definitely going to continue exploring the world of microbial genetics. There's still so much to discover.
Welcome back to our deep dive into microbial genetics. Are you ready for a few more thought-provoking questions?
Absolutely. I'm ready to push my understanding even further.
Okay, here's one that really makes you think about the connection between genotype and phenotype. Can two observably different cells have the same genotype? Explain.
Okay, so genotype versus phenotype. I think the answer is yes.
Okay, tell me more.
I think two cells can definitely have the same genotype, but have different phenotypes. The environment can play a big role in how those genes are actually expressed.
I agree.
Like, for example, let's say you've got two identical twin bacteria. They both carry the gene for producing a pigment that makes them appear red.
Okay, I like where you're going with this.
So if one twin is grown in a nutrient-rich environment, it might produce a ton of pigment and be bright red.
Right.
But if the other twin is grown in a nutrient-poor environment, it might not produce as much pigment and only appear pale pink.
That's a fantastic example. So even though those twins have the same genetic makeup, their appearance, their phenotype can be totally different just based on their environment. It's like two people with the same genes for height. One might grow taller if they have a healthy diet and plenty of exercise, while the other might be shorter if they don't have access to those same resources.
So it's almost like the genotype is the blueprint, but the phenotype is the actual building.
Yeah.
And the environment can influence the whole construction process.
That's a great way to think about it. Okay, let's move on to a question that really zones in on a key enzyme in DNA replication. Why is primus required for DNA replication?
Okay, let's see. Primus.
Yes.
That's the enzyme that makes those short RNA primers. Right?
You got it.
And DNA polymerase can't just start building a new DNA strand from scratch. It needs a starting point, like a free 3' hydroxyl group, to attach that first nucleotide to.
You are exactly right. And that's where primus swoops in to save the day. It provides that initial 3' OH group by laying down a short RNA primer, giving DNA polymerase a foundation to build upon.
So it's like primus is the construction crew that lays the foundation.
Yeah.
And then DNA polymerase can come in and actually build the rest of the house.
I like that analogy a lot. Okay. Let's switch gears just a little bit and think about a specific type of mutation.
Okay.
Describe what happens when a nonsense mutation is introduced into the gene encoding transposase within a transposon.
Okay, so transposase is the enzyme that allows those transposons to jump around in the genome.
Right.
And a nonsense mutation introduces a premature stop codon, which can really mess up a protein. So what would happen if a nonsense mutation occurred in that transposase gene? Well, if the transposase gene is cut short and non-functional because of that mutation, then the transposon wouldn't be able to move.
That is exactly right.
It'd be stuck, right?
It would be totally stuck in its current location, almost like, like a car with a broken engine.
Oh, yeah.
Unable to move to a new location.
So those jumping genes would be grounded.
That's a great way to put it.
Awesome. This has been so fascinating. I am really starting to appreciate how even the smallest changes in the DNA sequence can have a huge impact on the whole cellular process.
It's incredible, isn't it? And as we wrap up our deep dive into microbial genetics, I want to leave you with one final thought-provoking question. We've been focusing on bacteria this whole time, but how do you think gene regulation might work differently in more complex organisms like us?
Ooh, that's a good one. Well, I know that eukaryotic cells have a nucleus, which separates the processes of transcription and translation.
Right.
And their DNA is also packaged with these histones forming chromatin. I'm guessing all of these things add another layer of complexity to gene regulation. Like maybe there's more opportunities for fine-tuning gene expression in eukaryotes.
You're definitely on the right track. Eukaryotic gene regulation is like a whole other world just waiting to be explored. But for now, thank you so much for joining me on this deep dive into the amazing world of microbial genetics.
This was so cool. Thank you so much.
You've shown such a strong grasp of these key concepts and a real curiosity about these tiny wonders.
I've learned so much about how bacteria manage their genetic information, from DNA replication to gene regulation. I know I'm definitely going to keep exploring the world of microbial genetics. There's still so much to learn.
Absolutely. Well, that's all the time we have for today. Thanks for listening, everybody, and we'll catch you on the next deep dive.
Bye, everyone.